Hello and welcome to my exam mini video 4, where I'll be looking at DNA structure. So here we have a diagram of a nucleotide, starting at carbon 1 on the right. This is where our base attaches, and we have possible bases of purines, which are adenine and guanine, or pyrimidines, which is cytosine or thymine. Next is carbon 2. This is where the deoxy comes from in DNA. There's a lack of an OH group here that would be present in RNA. At carbon 3, that's what connects the phosphate group to the sugar group to form the phosphate backbone. And at carbon 5, we have our phosphate group. Here we have a diagram of base pairing. So we have hydrogen bonds between our base pairs. There's going to be 3 bonds if it's a GC pair, 2 bonds if it's an AT pair. So we have our phosphodiester bond that's created between the 3' carbon and the phosphate group to create our phosphate backbone of our DNA. The left side of the screen shows the 3' to 5' direction going from bottom to top. The right side shows the 3' to 5' direction going from top to bottom. That's what makes DNA antiparallel. So in this image, we're going to be looking at the minor groove towards the lower part of the screen, and then the major groove towards the upper part of the screen. The major groove, it is a lot larger than the minor groove, and that's how we can distinguish those. So we can see from this image that DNA is right-handed because the sugar phosphate backbone travels up and to the right. If it traveled up and to the left, it would be a left-handed. So now we're going to be looking at what salt concentration would do to DNA. So a higher salt concentration causes cations to shield the negatively charged phosphate groups. This eliminates the repulsive force between the 2 strands. So where the phosphate groups were pulling away from each other, creating some tension in the DNA, that repulsion is not going to be there anymore. So the salt concentration will lower the melting temperature of DNA. All of these effects could cause changes to the dissociation and base stacking, which are important factors in the stability of DNA. Thank you for watching. Here are my citations.